Um, let's go ahead and try to maybe record. All right. So the special senses is fascinating topic. We will discuss, mostly the vision, the taste, the hearing, and also the idea of the equilibrium. Okay. As far as the touch goes, we already covered the touch. We discussed the touch receptor, mechanoreceptor, proprioceptor, those things already in chapter 13. So as far as the chemical senses goes, there are chemoreceptors involved, obviously. So the chemoreceptors are involved. They detect chemicals in the environment. Okay, everything must be dissolved. So when we tasting something, like let's say something is salty, the salt should get dissolved in the saliva. Same thing with the smell. The chemoreceptor works best when there are detecting chemicals in the aqueous environment. Okay, so the smell is the same thing. All right, the substances must, dissolve in the fluid of the nasal membrane, whether it's your favorite aftershave or perfume or the smell of the flower or the food. All of those is enters the nasal cavity, and in there are a lot of mucus lining, which you learn in anatomy. They get dissolved in there, and then you have those olfactory receptors waiting, which again, if you reviewed earlier on chapter, I believe was chapter 11 at the end or 13, I can't recall quite well, but it's a G-link protein that is involved in the sense of the smell. So we'll take time and go over it really quickly and give you guys idea of what's going on. So as far as the taste goes, again, in anatomy, you learn that there are a lot of papillas on the surface of the tongue, and also there are a lot of taste buds. So we go ahead and take a look at a small section right over here. These are called papilla. Basically, they're finger-like projections. And on the side, right over here, you could see the taste buds. All right, the taste buds have been enlarged right over here. So one of these have been enlarged over here, so you could see it. And what do we see? We see gustatory hairs, which is another name for, the kind of like a part of a receptor. So you have gustatory receptor cells, which is our chemoreceptors. And there are other supporting cells around them. These supporting cells help out in terms of their structural ability and construction, as well as the stability of the cells. They're putting whole things together. Notice that there are, taste fibers. These taste fibers obviously are part of the cranial nerve. In other words, taking information. We discussed this already. Sensory neuron take information to the control center. In this case, would be the brain. All right. So these are that involved. So there are a lot of chemicals in our food. These chemicals in our food, which happen to be in the oral cavity, enters the taste pores. Then these taste chemoreceptors detect them, and then they send in the neurotransmitter and they communicate with these nerve cells. And these nerve cells are again are sensory neurons. Remember, we had receptors, which are this part right over here. Then you have the sensory neuron, which is this part right over here. But there are a lot of them that kind of like collectively they're going in towards the brain, which happen to be the control center. So first of all, this part right over here is the aqueous environment right here. That would be your saliva. Okay, so that's what we're going to get at. We go ahead and take a look at there are different types of taste sensation. Although this is very, very general, a lot more have been identified, but we're not going to get into it. Sweet, bitter, umami, those are all work via the G-link protein. Okay, so again, the others like umami, that kind of like beefy taste, or I don't know if you ever had umami burger and whatnot. You know, they have a unique taste. It's not really salty. It's not really bitter. It's not none of the above. So Japanese scientists discovered this, and they are elicited by the amino acid glutamate. As you know, the glutamate was one of those amino acids that was involved in the EPSP, excitatory postsynaptic potential. Anyways, so all of these, they're unique in a way that they have outside receptors, molecules or the chemical bind to the receptors. These receptors are linked to the G-link protein, and as a result of that, the G-link protein can, turn on a lot of relay molecules that we discussed in a previous lecture, including adenylate cyclase, hydrolysis of ADP to the cyclic AMP, and again, cyclic AMP can open up certain channels, and as a result of that, ions can go in and out. All right. However, the salt and the sour, they're really related to the cation. As far as the sour goes, that would be the hydrogen ion. As far as the salt goes, as you know, sodium chloride, they have ionic bonds between them. And again, when they are separated, they become Na plus and Cl minus. And this Na plus, due to the channels that exist there, can enter and cause the depolarization and therefore triggering the action potential, as you're familiar with. All right, so let's get to it really quickly. There are proposed mechanisms. So again, they must dissolve in the saliva. They must get picked up by the receptors. And after that, it's just the idea is the depolarization and releasing of the neurotransmitter. Neurotransmitter communicate with other neurons. Other neuron triggers their action potential. And as a result of that, that action potential is going to reach the brain, and that's what is happening. So that's the idea between pretty much most of these chemoreceptors that are involved. Now, how they generate action potential and release the neurotransmitter varies. They could be either due to the sodium and calcium or hydrogen ion, or they could be due to the G-link protein that we discussed. Let's go ahead and take a look. In a nutshell, that's what's going to be happening, and information from the various part of our tongue where the receptors are located, we'll take that information and we'll take it to the site of the particular part of the brain that you studied in anatomy, gustatory center, and then you'll be able to determine or interpret as this signal, what is the interpretation of this signal? Is it sweet? Is it salty? Whatever the case might be. All right. So again, you learn a lot of different type of, cranial nerve that are responsible for taste, movement of the tongue, things in that nature. I'm not going to go over those right now because you're supposed to have that already. So let's get to the travel inside of the tongue and take a look at the receptor cells. So earlier, I told you that these yellow ones are the receptor cells right over here. All right, you could see it. So if we go ahead and take this and kind of like show it to you, it'll look something like this. All right, pretend this is a yellow one. Okay, and again, if I upright it, then I could just draw it this way to make it simple. All right, these are the same thing. All right, these are the, this is the same thing as that one. This is the same thing as these yellow one, and there happen to be, receptor cells. So if we go ahead and just take a look at one of these on the next page, all right, perhaps, again, you could do this at home if you like to, you could map to see which type of receptors you have where. So for example, you don't need to know for the exam or the quizzes, but the tip of the tongue You could detect the sweet better, for example, all right, and salty on the side of the tongue. You could detect the sour, and the bitter you could detect towards the back of the tongue. So again, if you like to do these experiments, you could have a family member, for example, have your eyes closed and put in sugar molecule or salt or different type of chemicals in your tongue while your tongue is sticking out, all right, one at a time. And then you bring your tongue inside and you try to use your saliva to see if you could tell what it is, all right? So again, you kind of like could map these and you will find out the location in which those chemicals or substances are placed on your tongue varies. And some location you could detect it right away. All right. You put a drop of vinegar, for example, on the side of your tongue, you could probably tell right away versus the other one. Make sure if you do that kind of experiment at home for fun purposes, uh, to rinse your mouth with water every time you're doing this. Okay, so that's another criteria you could do. But let's go ahead and take a look at one of these receptor cells to see what it looks like. In this particular case, I wanted to show you guys because I've already done it. So this is what the salty tastes like. So sodium chloride in the saliva or water aqueous environment, they dissociate into sodium cation and the chloride anion. So this sodium cation is the important part. So there are protein channels just for sodium. So when they get dissociated, guess what? They could go right in. And because it's a cation, this could change the voltage of inside of the receptor cells, and that caused depolarization. This depolarization further down is going to open up various voltage gated channels, just like the neurons that we discussed, and it's going to reach towards the end where the calcium voltage sensitive channels can open and the calcium can come in. Now, this calcium is coming in, causing the exiting or exocytosis of the vesicles with the neurotransmitter, most likely glutamate. And this glutamate or the neurotransmitter that are releasing, again, bind to the ligand or chemical gated channel on the nearby neuron, which again, they trigger the action potential because it's excitatory, and then it sends a signal to the central nervous system. What if action potential did not fire? Then you wouldn't be able to taste that salt. There could be lower amount of salt in there, and as a result of that, lower amount of neurotransmitter being released, and as a result of that, that may not be enough to reach the threshold on these nerves. So these nerves are the sensory nerves, sensory neuron. All right. So earlier I told you in this diagram, these are the sensory neuron. So the cells that are in yellow, there happen to be the chemoreceptors that we discussed, and I just showed it to you separately. All right. So again, we have all these receptors all over your tongue. What about the other ones? Let's go ahead and take a look at the next one, which happened to be a detection of uh, sour materials, say vinegar or something that is sour, sour cherry, sour whatever. Okay, so most of the time is a hydrochloric acid that they dissociate into hydrogen ion and chloride ion. The concept is very much the same. Hydrogen ion channels are located on these chemoreceptors, and as a result of that, they could go right in, all right, through their channel. We discussed this was the facilitative diffusion. So when you have higher amount of hydrogen ion outside, they're going to go right in. Inside, they are negatively charged. They are cells. These are happen to be the chemoreceptors. So as the hydrogen ion comes in, that could reach the threshold right around negative 55 that we discussed. Action potential is going to trigger due to this depolarization, these action potential voltage differences is going to reach the calcium voltage gated channel, calcium moving in and causing the exocytosis of the neurotransmitter. Again, neurotransmitter communicate with the sensory neuron, which again communicate with the central nervous system or the brain, and the brain is the control center and it can interpret things. So sweet and sour are really simple. They just have their ion channels that they could go right in, and as a result of that, depolarization can achieve. What about the rest? The rest, whether it's sweet, whether it's bitter or umami or even other ones, now they're talking about other molecules and they are investigating it currently. They're all triggered by the G-link protein. The mechanism of it, there are several different proposed mechanism of it, but in general, I'm showing you what was talked about in your book. Again, if you read some certain articles and so on, there might be a slightly different according to different findings. But here it is, the sugar or the sweet sensation. When you're tasting a sugar is a glucose. So this is a glucose, for example. The glucose, they don't just go in. First of all, they're too large compared to the cation. All right. Another thing is they do not have the receptors in order to their protein, transport protein to get inside. So they bind to the receptor, and you could see these receptors are located right over here. So once they bind to the receptor of these cells, G protein gets activated. G protein can activate adenylate cyclase. All right, AD for adenylate cyclase, which again it can trigger hydrolysis of ATP. This is an enzyme that can break down the ATP to cyclic AMP. Cyclic AMP is the second messenger. In this case, this second messenger closing the potassium kind of like leak channels, so potassium no longer going out, and that causing the depolarization, which again, this depolarization, when it's great enough, is going to cause the action potential to fire, and the rest of the story is the same. All right? So just know that there is a G-link protein is involved. The cascade of the G-link protein, you should familiar with it as well. The mechanism over here, or proposed mechanism, there could be other mechanisms involved too. So just letting you know. Next up, we got the bitter taste. Bitter taste usually because of the different molecule. Quinine is one of those. If you have some quinine, you'll taste the bitter taste. Certain alcohol, not only they have sweet, but also they have a bitter taste to them. Certain beers, for example, and whatnot. But anyways, as the molecule dissolve in the saliva, they find themselves to their specific receptor. Again, this is the same idea, G-link protein, adenylate cyclase, cyclic AMP. And finally, this cyclic AMP can do different things. In this case, it can cause the smooth endoplasmic reticulum to release the calcium, which is sufficient enough to cause the exocytosis of these neurotransmitters. The rest of the story is the same. So in a nutshell, everything is involved with G-link protein except the salty, which is the sodium ion is involved, and the sour taste, which is the hydrogen ion is involved. All right. So that's as far as the taste I would like to talk about. We go straight to the olfactory senses or sense of smell.
Perfect.
Let me pause the video, see if you guys have a question, and then we can go ahead and talk a little bit about your question. Hold on 1 second. Go ahead.
Question, is calcium not involved in the sweet and umami taste? Are there no calcium voltage gates?
Let's see. On which one?
On the sweet and umami.
Calcium itself, could be involved because this is just telling you the depolarization, what happened with the depolarization. Depolarization could again open up the calcium channel. Calcium can enter and cause the exocytosis.
Okay.
Okay, so
Okay.
the story is very similar. Like I said before, there are different proposed mechanisms, but I just focus on the primary part of it, how this depolarization is going to be achieved.
Okay,
Does that
got it.
make sense?
Yes, thank you.
If you recall, the depolarization, once it occurs in the axon hillock, you had a lot of those sodium voltage gated channel, and then sodium would enter and cause the perhaps propagation or the action potential moving from one area to another. In here, of course, we do not have the axon hillock, but the propagation is very much similar. In other words, the depolarization is the first phase, but then the propagation of that is redundant. So I didn't talk about how the voltage gated channel can influence and refractory period and all that stuff that you already mastered. Okay? Any other question before we proceed? Okay. Let me go ahead and record the video again. Let me see what is on the chat. Huh? Okay. Not much important. If you're late, that's okay. Resume recording. All right. Next up, we want to go over the olfactory senses. Olfactory senses are really important. So, as you know, taste is 80% smell. All right. If for whatever reason you cannot smell, uh, food that you eat become very tasteless. People like I'm sure you have as well. You caught a cold at one point in time, and you can't smell things, and your appetite is being suppressed. You don't feel like eating anything or whatnot, and anything you eat, it just doesn't taste the same. All right. Uh, my advice to you: never take your friends out anywhere or invite them over if they are having a nasal congestion because they cannot appreciate the food that you prepare for them or whatnot. Okay. Uh, Nowadays with the COVID-19 is obviously there's not many people gathering to begin with, but interestingly enough, and maybe those people that are presenting about the COVID-19, maybe you could talk about why that the sense of a smell of certain patient disappears during that. Okay, but in general, there is a lot of mucus lining that they starts producing more and more mucus. That's why you get the congestion on the nose and so on. So preventing you from picking up molecules readily, and as a result of that, you can't smell well. All right. So that's the idea behind it. But a lot of things influence our taste, including thermoreceptors, which is again the ability of substances getting dissolved. It plays an important role. Having your ice cream while it's completely melted, it will taste very sweet because there's a lot of sugar now that are dissolved and so on. Mechanoreceptor, how you feel with your touch receptors on the surface of your tongue. You have nociceptors, these are for pain, again, something that is hot or spicy or perhaps, could create painful stimulation. So, temperature, they're all playing an important role, obviously, when it comes to the uh, taste. So we go ahead and take a look at the sense of smell. As you know, the sense of smell, uh, molecules, they travel inside of the nasal cavity. Right over here, this rectangular area have been enlarged in here, so you could see the mucus lining, all right, the mucus layer. So these molecules get dissolved right over here. And again, you could see the olfactory receptor cells right over here, right? They detect these. So these are also known as chemoreceptors. When we say chemoreceptors, it's just general terminology that they can detect chemicals in the fluid. All right, so right over here, they can detect these. Let's attach them to them and so on. And again, same ideas. By the G-link mechanism, the action potential is going to get triggered and is going to propagate along this, and again is going to communicate with other nerves, and these nerves obviously it's goes into the olfactory tract and part of the brain that is responsible for sensing the smell. Interestingly enough, our sense of the smell, it looks like through evolutionary course, have been declined. Well, why is that? I mean, when you're looking at some of the bone structure that we found on the early humanoid in the caves and so on, the nasal cavity uh, or the hole for the nasal cavity seemed to be a lot larger than what it is today, which suggests that we had better and larger nose and better capability of sensing different chemicals. Of course, for the survival, it was necessary to, find potential mates, find perhaps maybe food, and avoid predators. All of those was related for and use olfactory senses. But as the course of evolution took place, we became hunter and gatherers, and then pretty soon, humans were involved in trading and so on, and we seem to be using less and less of our olfactory senses. Don't get me wrong, it's still a lot of, in psychology, a lot of scientists believe that the odor plays an important role in terms of the attraction and so on. But our ability to pick up these molecules have been reduced over time. Right? Dogs, for example, they are much keener, and many other organisms to sense of smell is a lot more enhanced than ours. Uh, I was watching some time ago how a predatory cat, like tigers or lions, they attack uh, the herds or animals and so on, and they always go opposite side of the wind because the wind can carry the smell and the praise. They can sense that and get away, which I thought it was fascinating. That again tells you how sharp their sense of the smell is, and so on. Even sharks, they can smell the blood, for example, or other senses that they do have in the ocean. So there's really opening up a lot of different ways in which organisms use their olfactory senses. But let's get back to the ideas right over here. So what we're going to do, we're going to travel inside one of these olfactory receptors to see how the G-link protein is involved. And again, the idea is right over here. So this is olfactory receptor cells. All right, that's the membrane right over here, and this is the outside of the olfactory cells. All right, so this is the inside. All right. So as you can see, the odor chemicals are coming in and binding to the receptors. All right. Once it binds to the receptor, now it's bound to the receptor. So a little bit later time, this activates the G-link protein, which finally activates the adenylate cyclase, which finally, hydrolyzes the ATDV to the cyclic AMP. In this case, cyclic AMP opens up the sodium and the calcium channel, as you can see in here. Sodium for the red, for the yellow ones, and the calcium maybe we can put in red or we can put in a black one. Doesn't matter really. But both of these can go, and once the channels are open, they can just go right in. And as they go right in, this is positive ions, so it could cause the depolarization, and again triggers the action potential with propagation and so on that we discussed. The action potential can propagate through these receptors and then again release the neurotransmitters right around here, which again it can bind to the ligand gated channels, and then again communicate with other nerves that are going towards the olfactory tract and towards the brain. Okay, so pretty simple when you think about it. It's just G-link protein is involved, and you're familiar with it. This is just major mechanism involved. All right, so we're done with the taste, we're done with the olfactory. Let's go ahead and take a look at the eye. When it comes to the eye, our eyes are actually pretty good. 70% of all sensory receptors happen to be located in the eye. That means what? That means we use our eyes a lot for doing different tasks. Right now, you're using your eyes to looking at the screen and also listening. We'll talk about the sense of hearing later, but your eyes really, really important to bring in a lot of information from outside environment. Okay, there are a lot of accessory structure. These accessory structure help out. These includes the eyebrows, for example, that can provide some kind of a shade. Eyelids that can distribute the fluid so your eyes doesn't get dry. Also protect your eyes against the pollution, dust particles, things in that nature. Conjunctiva is a connective tissue, a layer that prevents microbes to enter and cause problems. Lacrimal apparatus that produces tears, again, constantly washes and moisten your eyes so there won't be any irritation. And if there is anything there, it could help to bring it out. And then also extrinsic eye muscles that you learn in anatomy enables you to move your eyes in different directions, left, right, up, depression, and elevation. All of those were the actions that you learned in the extrinsic eye muscle. Moving forward, we're going over in general, there were three layers of the eye. One of the layers on the outside was the fibrous tonic, which again was a sclera and connective tissue. The middle part was the vascular tonic, which is mostly choroid, blood vessels, and so on. And then the inner layer was the sensory tonic, and that would be the place where the photoreceptors were located. Those were all in anatomy. Nearby, there was a pigmented layer. It's important to know your anatomy, so review those if you can. And then once we get to the pigmented layer, you could see a lot of neural layers. There are nerves that are communicating with one another. Again, in anatomy, you learn that the photoreceptors were rods and cones, but there were also bipolar cells. There were also other cells known as horizontal and amacrine cells. Let's go ahead and look at it. So we're looking at the light as the light passes the lens and going towards the back or the surrounding or first layer of the eye, which is called retina. You could see the pigmented layer. Pigmented layer as they're made out of melanin. Melanin is a protein molecule that is darker, kind of like gives a color to the skin. And if you're wearing dark color and you're going outside, and today is pretty sunny, it absorbs all the light, so you get pretty hot if you're wearing dark clothes. Most of the people wearing lighter color in the hot days, like white t-shirt or pastel colors, things in that nature. So it doesn't absorb the light. Same thing in here. This pigmented layer absorbs the light. So as the light coming in, they get absorbed in the pigmented layer. Guess what? Nearby, there are rods and cones. We have more rods. Rods are responsible for distinguishing, very much seeing dim lights at nighttime, specifically. Rods are becoming really handy. You could pick up movement. These are peripheral vision related to the rods, and seeing different shade of uh, black and white, for example, gray color, things in that nature. Those are all the responsibility of the rods. We do, however, have cones. Majority of the cones are concentrated in the area that you learned in anatomy, and that would be fovea centralis, and where the macula is. And again, you might have heard about macular degeneration when you have a lot of damages to the cones, and as a result of that, you cannot see clear vision or central vision. Cones are starting with C, so the way that you could associate with this, clear vision starts with C as well, clear. Color vision, C. The genetic problems that I gave you guys related to the color blindness and so on, those are all the cones that are involved or malfunction. And also central vision. So they're triple C, central, clear, and color. Okay, so those are really essential as well. And by the way, make sure that you review the genetic, especially when it comes to the blood, whether it was a sickle cell, whether it was, blood typing, whether it was the genetics of the blood. Certain people with certain blood types, they are, let's say, reproducing. Make sure that you review the genetic parts as well. But you could also look at the guidelines. Anyway, so what's happening in here? Again, once the light picked up by the photoreceptors, they're releasing their neurotransmitters right here, and the neurotransmitter communicating with bipolar cells, which you could see it in the red color right over here. And again, other cells, specifically amacrine and the ganglion cells. And the ganglion cells, you could see they're kind of like bundling with one another, which together they go back up the eye, and that was bundle. All these bundles are known as the optic nerve. Optic nerve is basically the extension of all these axons of the ganglion cells collectively. And they exit through the eye, and again, they go to the occipital lobe where we have a visual association area, and you could determine whether you see color, whether you see movement, whether you see, peripheral vision, things in that nature. All right. Moving right along, if we go ahead and take a look at the photoreceptors, they're located everywhere. All these areas, they have photoreceptors. Again, think about three-dimensionally, except this part right over here. This part is called the optic disc or blind spot. We do not have any photoreceptors in here because this is for the passage of blood vessels and the nerves. So if the object falls in here, then you will not be able to see it. All right. So, however, we don't really feel it because the blind spot of one eye is covered by the visual field of the other eye. But if you had one eye to operate and so on, there will be some areas in which you may not be able to see, especially if the images fall in that direction. All right, so there are different other parts to it. We already went over the jobs of the rods and the cones. So I'm just going to close this and go to the next one. And that would be part 2. The photo transduction is rather complicated process. And again, you might be able to look at some of the YouTube video to get a better grasp of it. I'm just going to make it concise informative and yet easy to understand. All right. So inside of the rods and the cones, there are kind of like a coin-like structures. All right. These coin-like structures, they have plasma membrane that can change shape as they absorb light and offer a lot of things happening in those areas as well. So let's go ahead and take a look at it. So we are traveling inside of the, let's say, rods in this case, and inside of these, there are tiny little coin-like structures right over here. All right. And again, if you look at one of these and enlarge it, you could see the membrane in there, and you could see a special protein as well. Now, these proteins are known as opsin, but whenever another molecule bind to it, called retinol, then together they're known as rhodopsin. So you got opsin. When the retinol is not bind to it, it looks like retinol already bind to this one. So this is called a rhodopsin. What's the big deal? Well, here's what happens during when it's dark, all right, and when it's light. Two things happening, all right. They're different. So when it's dark, this opsin and retinol pretty much connected together, all right. So you have rhodopsin. However, when the light comes in, it changes the conformation of the retinol and no longer have the same shape. So that changing shape is going to cause a lot of changes. Let's go ahead and take a look at what happens, and then you guys can read the lecture on your own, as well as looking at the YouTube videos that I provided for you. So here is the retinol. Retinol is just a molecule. This retinol you could actually synthesize from vitamin A. So the carrot has good vitamin A's. It's a good idea to incorporate carrot in your diet to get these as well. Now this is called the rhodopsin. It's just two molecules joining together. And now there is no light coming in. We are in complete dark. But what happens if you go to a room and you turn on the lights? Switching the light is going to cause this. Remember, where are we at this point in time? We happen to be in the coin-like structures. The coin-like structure happen to be on the rods or the cones. So those are the photoreceptors. Photoreceptors happen to be near the pigmented layer. So when you turn on the light, it absorbs that light towards the pigmented areas. All right. Here is the pigmented area with the melanin that we discussed that has the darker areas, and they are right in the vicinity. So now this cone-like structure with rhodopsin, they can absorb the light, and as a result of that, it changes the shape of retinol. Bear with me. I know it could get a little bit complicated, but if you review it a couple of times, it's not as bad actually. So now the retinol and the opsin are separated from one another. What now? This opsin is kind of, not exactly, but it's kind of like a G-link protein. In other words, they are involved in a cascades of events. All right? There is a cascades of events taking place. All right? Which result in voltage differences. Not necessarily positive or depolarization, but voltage differences. In this case, hyperpolarization. Let's go ahead and take a look at what is happening. So light comes in, the retinol changes conformation. Now is more different looking. In this case, it was bent, but now it's no longer bent. So this activates the opsin. Now the opsin is activated, kind of like a G-link protein. It activates another molecule. This molecule is called transducin, which activates another molecule known as phosphodiesterase, PDE, which is an enzyme, kind of like adenylate cyclase. And what does this enzyme, phosphodiesterase, do? Phosphodiesterase detach the cyclic GMP. Now, cyclic GMP they are always attached to the certain sodium ion channels. All right, so a little bit, not a whole lot, little bit of sodium continuously coming in. That is during the dark era. When the light comes in and the cascades of event taking place, it remove and disattach the cyclic GMP, and then what happens? Sodium channel closes. Sodium can no longer go in, and so normally we discuss inside of the nerve was a negative 70 millivolts, but during the dark time, because the sodium keeps coming in a little bit at a time, we are looking at our resting potential at a negative 40. This is the reason why, because the sodium, a little bit at a time, it keeps coming in. Think of it as a leak channel, but these leak channels dependent on this molecule. All right, once it's attached to it, or think of it as a fancy hotel. They have a doorman or a nightclub or whatever. All right, when you coming in, somebody opens the door for you, or keep the door open for, customers. All right, holding on to the door so you could go in. But what happens when the light comes in? It's kind of like that doorman or that establishment is closed. No more anybody can come in. All right, so this is when this departs. When the doorman is gone, then nobody can enter. In this case, sodium ion cannot go in, and as a result of that, the negative 40 now becomes more hyperpolarized. Hyperpolarization causes because no more sodium is going in. Remember, we're putting a voltage meter in here and we're reading the voltage. So earlier, we are looking at the negative 40 millivolts. Why? Because of the sodium is keep coming in. So when this one is read at the negative 40 millivolts. But when you turn on the light comes in and all these cascades of events taking place. And as a result of that, this channel closes, no longer sodium can come in. Sodium is positive. So in this case, they're going to become more negative. So more negative means it has to be more than negative 40. In this case, it looks like they're going in beyond even negative 70. This is due to the fact that the sodium no longer is coming in. So look, action potential was a voltage differences. In our traditional action potential, we went from negative 70 to positive 35, and there was a voltage differences which translated to a signal. Same thing in here, except that this is a hyperpolarization. It still is a voltage differences that can translate to a signal. How they communicate with the other nerves and what kind of a neurotransmitters and so on, that one is a little bit more complicated. I could explain it, but I will leave it to you guys. If you are interested to know, I will maybe open up another lecture to give you just for your own knowledge and so on, but I'm not going to ask you that because it can get a little bit complex, and I'm not sure everybody in the class is going to be on the same level to understand what's going on. All right, so for that reason, all you really need to know at this point in time, just like action potential, that the depolarization or voltage changes was the signal. In this case, hyperpolarization or changing the voltage from negative 40 to more hyperpolarization is a signal as well. And this signal obviously is important in sending to the brain, which the visual association area is taking place. So these are called phototransduction. If you like, watch a couple of videos to see how it's done. Again, there are useful videos for you guys to look at as well. All right. All right. I'm going to pause the recording, give you guys a little bit time to stretch your legs, drink a little water if you like, and then when we come back, we'll finish this. We'll go over a few other things. We still have the hearing and equilibrium to go through, but for the majority, the vision is done, except the problem with the vision and also the accommodation and how our lens behave. But before we do that, go ahead and take your break. Come back 5-10 minutes, and then we'll carrier.